Hello, everyone. Welcome to this new session of Global IMMUNOTALK. So it's a great pleasure for me to introduce today Maria Casanova Aceves, who is our speaker at this major immunological event. So Maria is a junior group leader at the Spanish National Cancer Research Center in Madrid, in Spain, and there she leads the Cancer Immunity Lab. So she has been there since 2021. So she did her PhD in a very close institute, which is the Spanish National Research Center for Cardiovascular Diseases. And during her PhD, she worked in the aging and clearance from the blood circulation. And later, findings were found to regulate the physiology of hematopoietic metastasis-prone organs such as the bone marrow and the lung. So this was a very important discovery which pretty much opened a new research field within immunology and physiology. So empowered, Maria then obtained a Human Frontiers postdoctoral fellowship to move to do her postdoctoral stage at Mount Sinai Hospital in New York. So there she worked in the lab of Miriam Merad, and her project was to study the impact of lung-resident macrophages in cancer. So she showed that these cells were very important in supporting the epithelium to mesenchymal transition of tumor cells, and as well to support the early expansion of Tregs within the tumor microenvironment. So this really showed the amazing capacity of Maria to make important discoveries. So after her postdoc, she went back to Spain, where she is today, as I initially said. And today, her lab studies how micro and macro environmental factors shape myeloid cells in cancer. And her aim is to better understand what is going on, of course, with this disease, but also to open new therapeutic roads to treat it. So for this, she has obtained an ERC starting grant, and she has recently received a prestigious award from the European Macrophage and Dendritic Cell Society. Importantly, Maria So today, our special question for Maria is, Maria, if you could mentor your younger self, what would you say? Please, Maria, go ahead.
Thank you very much. Hello, everyone. I think my answer is that stay calm, things come. But I think I've always been running. So I think I can say that retrospectively, but it's very difficult to answer that question in time, which is something also I'm going to allude and talk about it during the seminar. So hopefully I can also train myself on now to say stay calm. You will see.
Great. Thank you, Maria. I think you can share your screen now and move on to the talk.
Okay, so can you see the slides? Can you see the slides? Okay. Hello, can you see the slides properly? Can you just do thumbs up?
It's
okay.
Good.
Perfect, Anna. Well, thank you so much for 2025 organizers to invite me to share the work we are doing in the lab. As Ana Maria explained, I've been studying myeloid cells throughout all my career, and I'm still very much interested in their biology, and I found fascinating, like every 15 days, every day, I learn something new. So I hope that you just enjoy the seminar, and also I invite you to ask many questions later on the YouTube channel. So the title of the seminar is From Flow to Faith, because I think that as an immunologist, and particularly a myeloid immunologist, I learned that I had to embrace flow cytometry, which is something that at the beginning I was not able to even understand. And then how over the years I've learned that both macrophages and neutrophils are able really to differentiate and change their fate, their function in tissues and more recently in tumors. So I hope I can walk you through all this knowledge I experienced over the years. And one of the things that I think I learned pretty early in studying myeloid cells is that the definition of these myeloid cells, it does not just rely on being a neutrophil, a macrophage, or a monocyte. It also requires their shaping by their circumstances or the niche signals that somehow remodel their function. So this really definition of nature versus nurture are really essential to understand myeloid cell biology. And just because I want to stratify things so that the concepts remain a little more clear, particularly for trainees, the nature of these myeloid cells, the most abundant myeloid cells, one of them are neutrophils. And what you can see here is just a cytospin, a very old technique that when hematologists apply to differentiate cells in a glass and under the microscope. And these neutrophils are very abundant, the most abundant myeloid subset in circulation in humans, and they are defined mostly, they were defined mostly because they were circulating and they were very, very far effective to fight against infection. They phagocytose bacteria, and by releasing their granules, their content, their DNA, they really can trap and impair, for example, impede systemic and sepsis responses, for example, upon bacterial infection. We can no longer define them as uniform, but when I started my PhD, that was the definition, how I define neutrophils. And actually, this was one slide of my PhD thesis. And something that I learned also in my PhD is that they are very short-lived cells. And this is very different to the nature of macrophages, which somehow have a higher degree of heterogeneity in tumors in tissues, when we're comparing with neutrophils. They have also different features, such as their time of residency and their origin, whether they arise from bone marrow progenitors or embryonic progenitors. And as I mentioned, they are extremely plastic at different levels, epigenetically, transcriptionally, and also very recently seen metabolically. So now, just that the nature of these cells has been defined, I'm going to walk you throughout many of the neutral mechanisms we are defining of these cells. And when you look at neutrophils, for example, they are kind of easy to define because what you see here, the granule, they have polymorphonuclei, they have also different granules, as you can see here by an electron microscopy image. And they are the vast majority of them defined just by the expression of a marker such as LY6E and CD11B. Here I'm showing you just an example of a plot of flow cytometry in which we have a model in which we can track the expression of neutrophils based on DS-REG. And what they do, as I mentioned before, is that they go into sites of injury or infections, and they really are able to do it this in a very, very fast and efficient manner, which is slightly different in the case of monocytes and macrophages. What you are seeing here is an intravital microscopy video in which this definition of a kind of behavior of neutrophils, which is named as swarming, helps also to trap bacteria and also to help remodeling the site of injury. So this is a degree of very fast effector cells. And what we discovered when I was doing my PhD is that this degree of life of cells that are both this degree of time of circulation in neutrophils occurs in a circadian manner. So what you are seeing here are plots of flow cytometry on neutrophils that they express two markers such as CD62L, also known as L-selectin, and CXCR4. And as long as the time of the day goes beyond, so what you see here is CT1, so it's 1 hour after the onset of the lights in the animal facility. This is noon, this is 4 p.m., 8 p.m., midnight, and 4 a.m. The levels of expression of these markers changes in neutrophils, indicating that at least in blood, there is certain degree of heterogeneity. So when we quantify these levels of neutrophils that we name as H neutrophils, because we were able to trace the time in which these cells have left the bone marrow and enter into the circulation, what we notice is that this population of red cells here that you see, that is this red square here, they circulate based on the time. So we could assess that this kind of behavior, the acquisition of this H phenotype, was circadianly regulated. And why they do that? Because we know now, years later, that when neutrophils stay a lot of time in circulation and they have this expression of CXCR4 high and they lose the expression of L-selectin, they become very reactive and they start to be very harmful for the vasculature. So on a daily basis, our body has generated a mechanism in which neutrophils are either produced and mobilized from the bone marrow into the circulation, or they are cleared and removed from the tissues, from the circulation, to avoid this collateral damage. And when they are clear, these neutrophils go into different tissues, right? That was something that it was not known before. But for the first time in the lab of Andres, we were able to identify that over 24 hours, doing parabiosis, so really differentiated neutrophils, that they have the expression of a fluorescent reporter, and they transmigrate into the tissues of this mouse that has not this color. So all the red neutrophils, they have to come from this mouse throughout the circulation that is established when you do parabiosis. We start to do flow cytometry and clear of tissues to identify these cells, and we identify that these neutrophils, they follow also this circadian infiltration in tissues across the entire body. We found these neutrophils that infiltrate that circadian manner in the bone marrow, in the spleen, in the lung, in the liver, in the intestine, and in the skin. And this, at this time, 2018, it was kind of very surprising because nobody was looking at neutrophils besides blood, bone marrow, spleen, or a little in the liver, right? So we found that somehow these neutrophils infiltrate. Some of them, they die there. Some of them return to circulation. But when they do, they leave certain messages, right? So we start to interrogate what were those messages. And for that, what we did is that we performed bulk transcriptomes of the tissues that were either infiltrated by neutrophils, that's what you are seeing here, by the PM, because normally neutrophils infiltrate the tissues at night in mice, starting at 8 p.m. And when we get rid of these neutrophils, we start to see that in the lung, we observe other changes, right? So really neutrophils, when they infiltrate, they were delivering messages into the physiology of this organ. And what I'm showing you here actually is the lung. So we start to trace kind of a map in tissues in which we could really identify diurnal or nocturnal modulation of physiology controlled uniquely by neutrophils. So you can see here, this is bulk RNA-seq of the lungs of mice infiltrated in the morning, so these time points, versus the ones that are infiltrated at night. So we could really anticipate on a time manner when the lung is infiltrated or not with neutrophils. So when we look, when we look at the processes that were enriched by this infiltration of neutrophils, and when we compare it with neutrophil deficient mice to know really which are the process that neutrophils control, we observe that when neutrophils infiltrated their tissues, they saw an enhancement these tissues gain certain functions, such as enhancement of ROS, changes in extracellular matrix, epithelial cell proliferation, and also enrichment of blood cell morphogenesis. So really showing that, besides the fact that neutrophils are preventing infections and ready to do, when nothing happens in steady state conditions, they are also controlling other functions, which Andres named as non-canonical functions of neutrophils. So we continue exploring those functions, trying to understand whether in tumors could also be taking some relevance. And what we notice is that when these neutrophils infiltrate the tumors, they can also enhance the production of reactive oxygen species. And this is the work that actually a very talented postdoc in the lab, together with two other fellows, Alba, is developing. It is really focusing on these circadian neutrophil side giver cues that neutrophils deliver into tumors. Maria is focusing on macrophages, and David is really the bioinformatician, which help us also to understand all the molecular regulation behind these changes. So as I was mentioned, when we analyze how these neutrophils might regulate also programs in tumors, what we notice is that neutrophils that infiltrate in the morning, they show an enhancement of ROX production, and consistent with our prediction in the bulk RNA-seq, we also observed that neutrophils that infiltrate the lungs in the morning, they saw an enhancement of proliferation of endothelial cells. This is dependent on neutrophils because when we get rid of them, we did not see these changes, and this doesn't occur in epithelial cells. So really supporting that neutrophil infiltration on a circadian manner can control time-dependent functions in the physiology of the lung, but also in tumors. And this is actually something that could explain this time preferential growth we have observed, not only in B16 melanoma tumors, but also in KP tumors, when we have engrafted them at different times of the day. And this is actually part of the work we are currently exploring, together with the help with a lot of histology analysis that Monica in the lab is doing. But there is also something apart in which, on a cell intrinsic manner, not only the message that neutrophils deliver into tissues, but the molecular machinery that BMAL1, which is the transcription factor that controls this circadian regulation, encompass in neutrophils. And what you're seeing here is how these neutrophils infiltrate the tumors on a daily basis, similar as what they do in steady state conditions. We have observed that these neutrophils infiltrate preferentially the tumors at night. We have also validated this on immunofluorescence. What you are seeing here is the staining of LY6G and these KP tumors that we fluorescently labeled with GFP expression. And what we are now trying to understand, which are the circadian programs that neutrophils actually are controlling in these tumors. And for that, what we are now starting to hypothesize is that, based on the tumor growth that I just showed you, that there might be windows in which these neutrophils, regardless of the abundance of these neutrophils in tumors, they might be inflicting actually more pro-tumoral functions, as observed by the preferential growth in the morning, whereas in the afternoon they might be controlling anti-tumoral functions. And this is something I hope I will be able to show you next year. Okay, but I told you that, neutrophils, okay, in blood, in tissues, they get a certain degree of signals. But first is where these cells are produced, right? And this comes again to the orthogonal integration of different determinants in neutrophil biology, particularly the origin, in which these cells are produced in the bone marrow through a series of progenitors. They get signals in the blood, which promote their clearance and their circadian aging, and then they get further determination in the tissues. So this bone marrow production can rely on different progenitors and generate different trajectories depending on the insult or the degrees of signals they get when a tumor grows. They can also rely on these daily fluctuations that I showed you before. And particularly, they can find an additional twist when tumors, for example, release different signals. And what you are seeing here is just a little snapshot of a fantastic work by Karen DeWieser, in which, using an amazing amount of genetical models, rely on different expression of oncogenes or tumor suppressor genes, she identified different levels of just neutrophils. But as you can see here, this is the quantity, and I showed you before that the quality requires further examination. So just analyzing the presence or the absence of P53, you can also anticipate a stratification in the molecular program that these neutrophils might be inflicting. Okay, So this is actually one of the questions that Mariola in the lab, together with Ale, a former postdoc in the lab, were really trying to identify. When and where neutrophil specification, this final twist in which neutrophils might either survive or die, for example, or generate nets or generate angiogenic programs, will be determined. And to answer that question, our study case are neutrophils in metastasis. So we take advantages of breast cancer models, and we look at how these changes in this primary tumor generate a different outcome in neutrophils which are associated to this metastasis. So we use different breast cancer models with different metastatic potential, and what we have seen is that neutrophils which infiltrate primary tumors, they upregulate a receptor that is named TREM1. And we do this in primary tumors generated either in genetic models, such as the polyoma T or the EU771, which is an orthotopic model with tropism throughout the lung. But when we look into this, into the expression of this receptor at different organs, generating experimental models of metastasis, we see that there is a significant enhancement of this receptor, particularly in metastatic sites such as the lung and in the liver. Interestingly, the degree of this expression is different on an organ basis. So really something also interesting to determine what is actually different to these organs. So we do this in the liver, and I show you what you are seeing here is flow cytometry plot in which we get neutrophils from either naive lungs or liver metastasis, and we just analyze the expression of this receptor TREM1. And as you can see here, almost a quarter of these neutrophils acquire the expression of this TREM1 receptor. So how is that occurring? We look into also human samples, and what you can see here is that when we look at organs that harbor primary tumors, such as non-small cell lung cancer or hepatocarcinoma, we are able to determine that the degrees of TREM1 can be either high or low. And this is quite specific of the neutrophil compartment, at least at the single cell level, although we are seeing also some macrophage TREM1 expression, as you might have seen around. So these levels of TREM1 determines like kind of a bimodal expression that could be used actually in advanced stage as an advanced stage tumor biomarker, and can actually report different biological states in tumors. So to further explore this, we take advantage of these public human data sets already published, and we start comparing what are the relationship between these TREM1 high and TREM1 low neutrophils on a tumor basis. So for that, we take advantage of these public data sets, and what we notice is that the expression of TREM1 highly correlates with the expression of mitochondrial genes, but also pro-survival genes, both in the lung and in the liver. Interestingly, something that we find pretty consistent in both organs is the presence of this PPIF, which I did not know what it was before, but what we now I know is that it's a component of the mitochondrial pore, which is open upon stress. Interestingly, when DNA from the mitochondria is released into the cell, there is also CGAS-STING signaling and inflammasome activation, which trigger the expression of inflammatory cytokines. So we are hypothesizing that actually TREM1 is the rheostat that facilitates that this pathway occurs. So to investigate this further, actually, we have generated a mouse model in which we have abrogated the expression of TREM1 using loxpiside and recombination driven by MRP8-3, which is a neutrophil-specific driver in collaboration with Andres Hidalgo, but originally developed by Emmanuel Passaguet from Columbia University. And what we have noticed is that when neutrophils lack the expression of these neutrophils enhance the expression of oxidative phosphorylation, as reported here by Seahorse analysis. As you can see here, a significant enhancement of basal respiration and maximal respiration as well. These neutrophils also that lack TREM1 expression, so enhancement of ROS expression, and not surprisingly, a deficiency into IL-1 beta production. Interestingly, we have not seen that these neutrophils have any kind of benefit from getting rid of this TREM1, which was something that somehow we thought might not be the case. But when we generate lung metastasis using full deficient TREM1 knockout, what we see is a significant reduction in these metastases. So really suggesting that TREM1 might be inducing actually this metabolic change that might induce a highly glycolytic environment supporting tumoral growth. We are also investigating how these TREM1 neutrophils interact with other compartments of the immune system in using maximum multiplex imaging. This is something part of the things we are interested in, how this TREM1 signaling actually is finally acquired in tumors. So let me now switch gears into how this determination that occurs in the tumors, that is modeled by the circadian time, by daily cues, can be also further remodeled by longitudinal cues. And this is quite relevant in the context of cancer, because what we know is that, particularly from the work I developed with Miriam, is that when tumors arise in a tissue, they might be growing together with certain cells that were already in the tissue, as opposed that when these tumors are already in a very late stage that really require the support or the help of additional cells that infiltrate the tissue, but they were not before when this oncogenic twist arises for the first time. This is something that we have discussed extensively in a recent work we published together with two very talented members of my lab, Jan Fogstad and Sarai, a postdoc in the lab, in which we put together different determinants that can really generate this kind of portal and imprint macrophage heterogeneity. So now we are going to focus on how this time ontogeny can really play a role when tumors are initiated. Okay, you might remember the work that I developed with Miriam, in which these early initiation events are pretty much controlled by this tissue resident compartment, and thinking that they are forming an alveoli, they generate this kind of EMT invasive processes, but they also, by the expression of TGF-beta, are able to generate early suppression mechanisms by the expansion of these early regulatory T cells. But later on, this, and depending on the model you are using, actually, these alveolar macrophages also can very much control dormant mechanisms when we analyze how, for example, breast cancer cells occupy the niches. And this is the work we developed together with the lab of Julia Virreguiso, now at Albert Einstein in New York. So as you can see here, depending on the model, the same cells that are in the tissue, so resident macrophages, can either generate EMT process, migration process, or stop these cancer cells and help them to remain dormant for many, many years, which actually is kind of a really clinical and relevant question that, particularly in breast cancer, many clinicians are trying to address. But what does it happen when these early alveolar macrophages start to be like dysfunctional and generate these kind of preterms in over tumor progression? What we notice is that as long as these macrophages reorganize, they promote also the activation of certain cells, which are fibronectin and alpha SMA positive cells. So we name them as cancer-associated fibroblasts. And concomitantly to this activation, there is an exclusion of these T cells from the tumor beds. So how can we connect these three events experimentally? So for this, we gather forces, Eduardo, a very talented PhD in the lab, together with Anna, a postdoc that recently joined the lab, and we start to interrogate which might be these kind of structural changes that occur and are controlled by different types of macrophages in lung cancer. And for that, we took an unbiased approach, and the first thing we do is we perform single-cell transcriptomics both in the leukocytic compartment, CD45, and CD45 negative compartment, either using mice which were depleted of the resident compartment, CD169 DPR mice, or depleted of the infiltrating macrophage compartment using CCR2 knockout mice. And surprisingly, when we did that and we generate this huge data set of single cell, what we noticed is that the compartment that was mostly impacted by the presence of different types of macrophages, which were the CD45 negative compartment, so to speak, the stroma compartment. So looking closer, then when we integrate all this data set from single cell, what we notice is that this population here that corresponds to this mesenchyma, mesothelial and fibroblast compartment was indeed the most significantly impacted. And what you are seeing here is the heat map of the different compartments that we noticed that were like just very condensed in these two populations here. So this mesothelial, universal, activated fibroblast, and this other type of activated fibroblast. When we start quantifying how these cells were present in the different macrophage mice, what we notice is that when we lack this resident compartment that we have depleted before, transferring our cancer cells, what we notice is that there was an expansion of this universal fate, but more interestingly, in the tumors, we observe a completely and significant depletion of this subset of fibroblasts activated. Indeed, when we validate these single cell experiments using flow cytometry with given markers to determine these populations using flow, what you can see is that tumor mice that were engrafted either in the absence of CD169 macrophages or in the absence of monocyte-infiltrated macrophages, we notice a significant expansion of this universal fibroblast and expansion as well of this PDGFR photoplanin negative population, and a significant decrease of this double negative population, which has been shown in the literature of many fibroblast experts, such as cancer-associated fibroblasts. So really showing that CD169 macrophages are the ones that can control the distribution of fibroblast fates in tumors. Next, we wanted to identify how can we use actually bona fide markers for this population. And with this, we gathered forces together with Shannon Charley, previously at Genentech, that she was very nice and helped us to really fade map this population of fibroblasts. So using DPT-CRT model, we track the fate of this marker of these fibroblasts, and we notice that, as you can see here, both in confocal imaging and by flow cytometry, indeed there is a population of cancer-associated fibroblasts which express DPT and that populates tumors very early. We continue investigating how these fibroblasts from DPT origin were generated, and for that we use pseudotime analysis. As you can see here, this universal population of fibroblasts is able to give rise both to activated fibroblast 1 and activated fibroblast 2, which is actually the one that is mostly controlled by CD169 macrophages. Then we generate a mouse model in which we backcross this DPT, this bona fide mouse model of a universal fibroblast. And when we deplete CD169 macrophages, what we notice, similar to what we did at without this reporting, we notice a significant reduction in the cancer-associated fibroblasts, concomitantly an expansion of this universal fibroblasts. So really showing that these macrophages can control the conversion of these universal and homeostatic fibroblasts into a more cancer-associated fibroblast. So what are these cells doing? Actually, these universal fibroblasts are very much very much related to the control of extracellular matrix and collagen deposition. And as you can see here, when we get rid of these macrophages, not only these tumors are smaller, but we also notice that the activation levels were very much decreased by the staining of alpha SMA, but also using different activation and cancer-associated fibroblast markers in the different models. So as you can see here, when we get rid of these macrophages, we observe that markers such as NMPTNT, alpha-SMA, TGF-beta, tenecin were significantly reduced, and this did not occur when we get rid of these monocyte-derived macrophages. So showing the specificity of this priming of universal fibroblasts by the population of resident max. Interestingly, this resident max also controlled the permeability of the organ, and this is shown here, in which Edu did an experiment, just very, very cool and simple experiment in which just using tumor-bearing mice, in which he has either depleted these macrophages or abrogated the infiltration on the monocyte macrophage compartment, he showed that really, in the absence of these macrophages, the permeability of tumors is further enhanced. Okay, but what are the consequences of having identified this crosstalk? As I showed you before, as long as this tumor progresses, there is an activation of these fibroblasts and an exclusion of the T cells in the body of the tumors. So the benefit of getting rid of these macrophages is shown here by survival, and this survival is not advantageous when we compare with mice that have been depleted of the monocyte-derived compartment. But interestingly, what we saw is that when these macrophages are not present in the tumors, we see a massive infiltration of CD4 T cells and CD8 T cells. And this does not occur when we get rid of these macrophages once the tumor has already been generated. So really suggesting that the priming of these fibroblasts into the cap phase is responsible for the exclusion of the T cells very early in tumor progression. So when we look into the profile of the T cells, what we notice is that these T cells actually, when we get primed by these cancer-associated fibroblasts, they generate a way more exhaustion phenotype. However, when we get rid of these macrophages, this exhaustion profile of T cells, that believe me, you see it here in this red ball here, is not longer present. So really showing that the acquisition of the CAF phenotype boosts exhaustion in T cells, and this can be redeemed or reversed when we get rid of these macrophages. And why is this cool? Because then when we combine this with immune checkpoint blockade, actually we observe a massive beneficial effect in the therapeutic setting and the survival of these mice. So really showing that these early changes can determine either response or efficacy, improve efficacy in immune checkpoint blockade settings. Okay, so another layer of complexity we are studying in the lab is how clonality might determine different subsets of myeloid cells. And I'm showing you here just a snapshot of the review I mentioned before from Jan and Sarai, in which we discuss about how these macrophages might be of different flavor or might acquire these early determinants that I was mentioning before, either depending on the progenitor in which they arise, either whether they arise from embryonic hematopoiesis or from adult hematopoiesis. And this has also very nicely recently seen by the lab of Stefan Jan. So Sarai is working in understanding something that is kind of the paradigm of clonal hematopoiesis, which is the presence of this clonal hematopoiesis of indeterminate potential in tumors, which I name as not so long indeterminate, because we are really starting to gain more insight into what are these cells doing in tumors. So as you can see here, hematopoiesis normally follows kind of a hierarchical structure in which hematopoietic stem cells are renewed and throughout a series of differentiated progenitors give rise to all the cells from the circulatory system. However, when you get into an event of clonal hematopoiesis, there are given mutations that normally accumulate during aging, but also about some stress, as was seen by bioRxiv publication in the first responders of 9/11. And these somatic mutations can be accumulated and inherited in the different progenitors, which ultimately lead to an expansion of myeloid cells, which are going to be populating your circulatory and your tissues. So this clonal hematopoiesis of indeterminate potential has been very much linked to age. but also to the myeloid skewing bias and to the presence of very much inflammatory circuits that can also boost and foster an ideal pre-cancer phase for cancer cells to expand. So we decided to focus on how these chips might be relevant in high-grade serous ovarian cancer because we identify certain data sets in which we observe a lot of presence of these chip mutations also associated to DNA and chemotherapy. So to start investigating this, what Sarai set up is a model in which we try to emulate how this clonal hematopoiesis occurs in human, and for that we set up a bone marrow transplant in which we irradiate host wild-type mice, and then we transfer a mix of bone marrow cells either from wild-type tumors, from wild-type bone marrow, or from TET2 deficient mice, which is one of the mutations most abundant in clonal hematopoiesis. And by doing so, together with Teresa, a very talented PhD in the lab, they are all talented, but I really believe they are all talented to be. What we noticed is that there is a very fast expansion of these clones, as you can see here, when we account for the expression of TD tomato in these chip mutated cells. We observe only just by 8 weeks, like a massive expansion, particularly in myeloid cells. So when we challenge these chimeras with ovarian tumors that we inject intrabursally in ovarian, in the ovary, what we notice is that these tumors grow massively, as you can see here, but also that there is a massive expansion of macrophages here, as you can see by immunohistochemistry imaging of F480. And these tumors are very much depleted of CD4-8 and CD4 T cells. So we are now currently investigating what is the landscape of these macrophages in TET2 tumors. And as you can see here, this is the significant expansion of macrophage phenotypes we see. I've never seen such a party of macrophage phenotypes in many years. So I'm very, very excited to understand this further. So this was a very long seminar full of data, but I think that the main thing that I want to conclude today with all the determinants we are exploring in the lab is that immunity and myeloid immunity is not static. It's a story that is very much written in time, space, and tissue and tumors, as I showed you today. And I think that the only way we will be able to understand and treat cancer, in particular to manipulate these cells, which is actually the focus of my lab, is embracing systemic responses, but also risk determinants to really decode, probably with AI, this dynamic intelligence of the immune system that keep us alive. So thank you very much for your attention, and I think I will be happy to answer all your questions throughout YouTube.
Thank you very much, Maria, for this fantastic talk. Oh,
this is the most this is the most important slide, sorry, which is, of course, all the people that make the lab possible with all these projects. Super cool, the funding agencies and our collaborators. Thank you, sorry.
No problem. So thank you, Maria, for this wonderful talk, for sharing exciting science with us, your exciting science with us. So I remind to everyone that you can use this QR code and the YouTube channel in order to ask all the questions you have to Maria. And I will also not forget this week to announce the next week's speaker, who is Daniel Saban. Don't miss him. Thank you, all of you. Thank you, Maria. Have a nice evening or day, depending on, or night even, depending on where you are. Bye-bye.
Thank you. Bye.